Welcome to G Fun Facts Online. Today we're looking at a disease that many of us fear, Parkinson's. For decades, we've thought of it as a kind of murder mystery inside the brain. An unknown assailant, maybe a mix of bad genes and environmental toxins, starts killing off vital brain cells. But what if that's not the whole story? What if it's less of a murder and more of a catastrophic burnout? A story of the brain's most dedicated workers being pushed to their absolute breaking point and beyond. Today, we're exploring the compelling theory of dopamine burnout, a new perspective that reframes our entire understanding of Parkinson's disease. Let's get into it. The classic story of Parkinson's centers on the death of a very specific group of brain cells, the dopamine-producing neurons located in a small, dark strip of tissue in the midbrain called the substantia nigra. These neurons are the master controllers of our voluntary movement. When they die, the brain can't produce enough dopamine, and the classic symptoms appear: tremors, stiffness, slowed movement, and problems with balance. By the time someone notices these symptoms, it's a grim reality. An estimated 60 to 80% of these crucial cells are already gone. For years, scientists have focused on the crime scene evidence. The number one suspect is a protein called alpha-synuclein. In a healthy brain, it's a normal, unassuming protein. But in Parkinson's, it misfolds and clumps together into toxic aggregates called Lewy bodies. The theory is that these clumps spread like a contagion from cell to cell, gumming up the works and triggering cell death. The other key pieces of evidence are mitochondrial dysfunction and oxidative stress. Think of mitochondria as the tiny power plants inside every cell. In the dopamine neurons of Parkinson's patients, these power plants are consistently faulty. They fail to produce enough energy, and, worse, they start churning out toxic byproducts called reactive oxygen species, or ROS. This creates a state of intense oxidative stress. The very process of making and using dopamine is messy and generates its own oxidative stress, so when you add failing mitochondria to the mix, you get a vicious cycle of damage. The cell's internal structures are corroded from the inside out, leading to its ultimate demise. Now, these mechanisms are all real. We see the Lewy bodies, the failing mitochondria, the oxidative damage. They are the undeniable fingerprints at the scene of the crime. But this classic view has always left a nagging question unanswered. Why these specific neurons? Why the dopamine neurons in the substantia nigra? The brain has many different types of neurons. It even has other dopamine neurons in a neighboring region called the ventral tegmental area, or VTA, which are largely spared in Parkinson's. So why this one specific population? What makes them so exquisitely vulnerable? The answer, according to the dopamine burnout theory, isn't about an external attacker. It's about the intrinsic nature of the cells themselves. This theory suggests that the decay isn't a passive process of succumbing to toxins, but an active, catastrophic failure driven by the cell's own desperate attempt to keep functioning. It proposes that these neurons are, by their very design, natural-born workaholics, living on a perpetual metabolic knife edge. Let's break down what makes them so unique. First, they have vast and complex structures. A single dopamine neuron in the substantia nigra can have an incredibly long and highly branched axon, forming hundreds of thousands of connections, or synapses, throughout the brain. Just maintaining this enormous physical network requires a colossal amount of energy. Second, they are pacemakers. This means they fire electrical signals spontaneously and continuously. Even when they're not receiving any input from other neurons, they are always on. This constant firing is driven by a steady influx of calcium ions into the cell. And third, because of their massive size and their constant, self-driven activity, these neurons have an exceptionally high metabolic demand. They are constantly burning fuel in their mitochondria to power their endless work. Think of them not as standard sedans, but as high-performance Formula One engines that are forced to run at maximum RPMs, 24 hours a day, 7 days a week. Even under normal healthy conditions, they are operating close to their absolute limit. They have very little reserve capacity to handle any additional stress. So you have these cells that are already running hot. Now let's introduce the first problem. This could be a genetic mutation, exposure to an environmental toxin like a pesticide, or just the cumulative wear and tear of aging. This initial insult causes a few of the dopamine neurons to malfunction and die. This is where the brain's amazing ability to compensate kicks in. To maintain normal movement and keep dopamine levels stable, the remaining healthy neurons pick up the slack. They ramp up their activity, increasing their firing rate and pumping out more dopamine to cover for their lost comrades. This is a brilliant short-term solution, but it has devastating long-term consequences. It's like a factory where the workforce is shrinking, so the remaining workers are forced to work double, then triple shifts, indefinitely, without a break. They can keep production up for a while, but eventually they will burn out and collapse. This is precisely what the burnout hypothesis suggests happens at the cellular level. This state of chronic, forced overactivation triggers a self-destructive cascade. The increased firing rate leads to a massive, sustained influx of calcium ions. While calcium is vital for cell signaling, too much of it is profoundly toxic. The cell's internal systems for managing calcium are completely overwhelmed. The mitochondria, the cell's power plants, work overtime trying to absorb this excess calcium, but this heroic effort cripples their main job, producing energy. With the mitochondria compromised, the neuron's energy demands, which are already skyrocketing from the hyperactivity, cannot be met. The cell is pushed into a full-blown energy crisis. The high-energy state paradoxically makes the mitochondria less resilient and more prone to damage. They begin to fail, leading to a massive surge in the production of those harmful reactive oxygen species we talked about earlier. This unleashes a firestorm of oxidative stress. It's a perfect storm. The metabolism of all the extra dopamine being produced also generates oxidative stress. This, combined with the failing mitochondria, creates a level of damage that the cell's antioxidant defenses simply cannot handle. This toxic environment is the perfect breeding ground for the famous alpha-synuclein clumps. The oxidative stress can damage the protein, causing it to misfold. The high level of neuronal activity itself has been shown to increase the release and aggregation of alpha-synuclein. In turn, these toxic protein clumps further damage the mitochondria, creating a deadly feedback loop that accelerates the entire death spiral. The burnout theory beautifully connects all the dots. The overactivation leads to calcium overload and metabolic collapse, which causes the oxidative stress, which in turn causes the alpha-synuclein to misfold and aggregate. It's not a mystery attacker, it's a system being pushed into a self-destructive frenzy. One fascinating part of this model is that the neuron starts to fail at its connections, its synapses, long before the main cell body dies. In a desperate attempt to protect itself from the toxic overload, the neuron may actually start shutting down its own dopamine production. It essentially burns out its connections before the cell body finally gives up and succumbs to programmed cell death. This new perspective is more than just an elegant explanation. It opens up exciting new avenues for treatment. If chronic overactivation is the key driver, then maybe we don't have to just focus on replacing the lost dopamine with drugs like levodopa. Maybe we can find ways to protect the neurons by calming them down before they burn out. This has led to research into therapies that directly target this hyperactivity. One logical approach was to block the specific calcium channels that drive the constant pacemaking of these neurons. A class of blood pressure drugs called calcium channel blockers, specifically one named isradipine, was tested in large-scale clinical trials. The idea was to gently tap the brakes on these overactive cells, easing their metabolic burden. While the trial ultimately didn't show a significant effect in slowing the disease, the core idea remains compelling. It's possible the dosage wasn't quite right, or that a different drug targeting these channels might work. Science is a process of refinement, and this result doesn't kill the theory. Other strategies focus on reducing the metabolic stress directly. This could involve therapies to improve mitochondrial health, boost the cell's natural antioxidant defenses, or even provide neurons with alternative energy sources so they aren't so reliant on their overtaxed mitochondria. Finally, technologies like deep brain stimulation, which is already used to manage motor symptoms in later stages, could potentially be adapted. Perhaps, if used early in the disease, it could help modulate and normalize the firing patterns of these vulnerable neurons, preventing them from ever entering the burnout phase. The dopamine burnout theory represents a major evolution in how we see Parkinson's disease. It shifts the narrative from a simple case of cell death to the tragic internal struggle of a cell population with an inherent vulnerability, pushed into a fatal frenzy by its own essential functions. By understanding this, we gain new targets and new hope. The goal is to move beyond managing symptoms and toward developing strategies that can protect these precious, hardworking neurons from their own exhaustive efforts. And finally, slow or even stop the relentless progression of this devastating disease.